Welcome to Genetics Lab in the fall of 2020. My name's Shane Breheny, and me and my partner, Courtney, are studying Tetrahymena. So Tetrahymena is a single-celled eukaryote found in most freshwater environments. They're easy to grow. Like some organisms, they're covered in cilia that provide for motility and manipulation of food for ingestion.
Tetrahymena are not the only organism that we know of that have cilia. In fact, they're quite abundant. They can even be found in the human body. And many organisms have other appendages, such as flagella, that are important for a variety of biological processes. But we know that Tetrahymena do have cilia, a lot of them, and that makes them useful for studying the structure. Tetrahymena have a relatively short life cycle. They are relatively inexpensive and cost-effective to handle in a laboratory, and they have a well understood genetic code, which make them a very ideal model organism for this type of genetic molecular research that we are engaging in. In Tetrahymena, the cilia are arranged in structures called axonemes. These axonemes are composed of 20 microtubules. As you can see in this figure, there are 9 pairs of outer doublet microtubules and 2 central microtubules. The outer doublets are connected by dynein, which are motor proteins, dynein motor proteins. As you can see, they're labeled here as dynein arms. And dyneins are expressed elsewhere in the organism. These ones, because they're found in the axoneme, are referred to as axonemal dyneins. That should go without saying. But these dyneins are really important for ciliar movement. The contraction of the dyneins causes the microtubules to slide, and that is how the cilia move and the cell gets about. And these dynein proteins that play a big role in the ciliar movement are multi-subunit proteins, as you can see here. There is all these different shapes in this representation. Could be seen as different subunits. We have heavy chains, light intermediate chains, and light chains. And these are all parts of the dynein protein. And for this specific project, we are focusing on a light chain protein that comes from the gene LC4A. Our plan for this project is to disrupt that LC4A gene product and test the effects of it on Tetrahymena. Other mutations have been known to drastically alter the swimming speed of Tetrahymena, but the LC4A gene product has not been characterized yet. So that was our goal for this project.
So our goal for the experiment was to disrupt the gene LC4A. We must do a few things first. First, we have to isolate the Tetrahymena DNA, amplify the gene of interest, and then transform the gene into an E. coli. First, we need to isolate the DNA. This allows us to ensure that any result we receive in the future is a result of the genetic mutation and is not affected by any other cellular components. The isolation of the DNA also allows us to perform our next step, which is to amplify our gene of interest. So the 3 main steps to amplify our gene of interest is number 1, primer design, which designates this gene as the one we want, LC4A, for amplification. PCR is the actual amplification. This makes many copies of the gene that we want to study. Agarose gel electrophoresis allows us to verify that we've amplified a gene and that we can advance.
So getting into the specifics of how we amplified our gene, primer design is a really important step. It's necessary for the polymerase chain reaction to occur properly and amplify the gene that we want. We used some pretty cool technologies like the Tetrahymena genome database and a website called Primer3 that made the primer design process very easy and automated. We just identified the gene that we wanted to amplify, and those programs gave us sequences that we could generate primers with. And these primers, essentially what they're doing is they're telling the PCR machine which gene to replicate, and we will see a little more about that in the PCR step. So this diagram kind of shows an overview of PCR, really simplifies it. So step 1, we have our DNA. that we inserted into our solution, and that DNA was also in combination with our forward and reverse primers that we identified in the primer design step, as well as some other chemicals that help the reaction run correctly. But the actual polymerase chain reaction machine, what it does is it will denature our DNA, and then the primers will anneal to the DNA. And so the primers that we designated or that we designed are complementary to the sequences of the gene that we want to designate, and they're actually the region that we are replicating is a little bit larger than the gene itself because we wouldn't want to leave it short. We want to make sure we actually get the entire gene. And so annealing is just the attachment of those primers to the designated spots that they're supposed to attach to. And these are necessary for replication, as we've learned in DNA replication. And so by only including primers for the sequence we want to replicate, which in our case includes the LC4A gene, we can ensure that only that sequence and gene will be replicated because those are the only primers that we've added to the solution. Then the replication step is really what happens in the PCR machine. We don't add anything special for that. We just simply provide the conditions necessary for that replication to occur. And that step is repeated many times, which provides us with so many copies of the gene that we wanted to amplify. And that's why it's called amplification, because the repetitive replication really almost blows that gene up for us. So after we've run our polymerase chain reaction, we can sort of check our results using a technology known as agarose gel electrophoresis. Basically what this does is it separates our products out, and by size, it will give us this rough size of our PCR product. And we kind of have an idea of how big we would expect our amplified gene to be. And so we can use agarose gel electrophoresis to kind of verify that we've created a product conducive to moving on with the experiment. And so this is an example gel. This is not indicative of our results, but it does outline the important parts of the gel and allows me to explain how it works. So over here on the left, we have this M stands for marker. This is where we insert a marker, which acts as a legend or a key. So these numbers correspond to general sizes, and we can compare the sizes of those markers to the sizes of our product. And if it's close to what we would expect, we can kind of deduce that our PCR was run correctly and we can move on with the experiment. And so, for example, we would insert our product into, say, well 1. And as we can see, the product of well 1 is a little under 700. Let's go 650. And if that's kind of in the ballpark of what we would expect, then it's reasonable to say that our PCR was correct, run correctly, it was successful, and we can continue with the project. And so the next real big step, this is just an overview. What we want to do is, take our gene that we've amplified and transform that into E. coli so that eventually we can make knockouts, disrupt the gene that we just amplified, and study its effects. And to do that, we first have to do a ligation. So what ligation is going to do is combine our insert, which is our PCR product, with a vector. And that creates a plasmid. And what we're going to do is transform that plasmid into competent E. coli cells to eventually create a knockout. We'll get to a little bit more about that transformation later. But what's important about the ligation is that, one, the vector was cut at the LAC-Z gene. LAC-Z is a bacterial gene that for blue and white colony screening that we're going to be doing later is very important. So this is really common protocol, and we'll get to why it's important later. And the other important aspect of our ligation is that we combined insert and vector at a ratio of 3:1. So there's much more insert than vector in our solution that we're going to use for the ligation reaction. And the reason this is important is because in order for this plasmid over here to be created, the insert and the vector have to not only collide in solution, but collide in the correct orientation. And that's how any sort of chemical reaction works. By loading up our ligation reaction with a lot more insert than vector, it really gives us optimal chances of that correct collision happening to form this full plasmid. And we really don't want to run this reaction and not end up with enough plasmid because it could certainly affect our results later in the project. So after we've performed the ligation, what we're going to do is a transformation where we take our plasmid and mix it with competent E. coli cells so that it can eventually be incorporated into their genome. Competent E. coli cells have alterations to their cell wall that makes it easier for foreign DNA to enter. So that's a really key part of the transformation that allows this project to be successful. And what we would expect to result from this transformation after we plated these competent E. coli cells with the foreign DNA incorporated is that we would see white colonies and blue colonies. And this goes back to the idea of cutting our vector at the LAC-Z gene. The LAC-Z codes for beta-galactosidase, enzyme that's really studied popularly in bacterial gene expression. But what happens is essentially the recombinant DNA, which is the DNA that has incorporated our PCR product, our insert, that plasmid has been incorporated. Those colonies are going to come out to be blue on the plate, and those colonies that have not incorporated our product are going to come out to be white. And so then we can just simply pick our blue colonies and continue with the project from there. So it's another step that really verifies the work we've done so far and helps us ensure that we can continue and we wouldn't be wasting our time. And so that comes to the end of the work that we performed this semester. Now, I mentioned a couple times throughout the presentation that we wanted to create knockouts of tetrahymena with a disrupted LC4A gene. Now, we at Simpson College aren't going to do this. What we're going to do is send our recombinant DNA from those E. coli cells to a place called the Tetrahymena Stock Center. And what they do is they specialize in tetrahymena genetic work, and they're going to create the knockouts for us and send them back because it's a process that we're not really capable of performing here at Simpson. But once we receive those knockouts back, Simpson College students will be working again on this project in cell biology lab. What they're going to be doing is phenotypic analyses of these knockouts. So that's where they're going to actually study the effects of what we've done in genetics and molecular biology. And, you know, unless you work at the Tetrahymena Stock Center, you might kind of be thinking at this point that this seems a little pointless, right? Who cares about cilia and tetrahymena and altering those genes? Well, cilia is actually all around us, including inside our body. This picture, you can read this, really shows a specific example of cilia in our body, but they can be found in the lungs. They really help us not to inhale debris or other harmful particles. They will trap those particles. That's the role of the cilia in the bronchi. And they can also be found elsewhere in the body. But if our cilia are beating abnormally, it makes sense if they're found in the lungs that could cause respiratory disorders. And it can also cause a condition called hydrocephalus, which can really have an array of other negative effects. So abnormal ciliary beating, which is what we would expect from our disrupted LC4A gene, if that happens in the human body, can really have some negative effects. And so that is one reason that we consider this research pretty significant for areas outside of tetrahymena itself. That's really the summary of what we've done this semester, and I hope you learned something. Thank you for listening.